So we are on chapter 9, section 5, and we're going to talk about that end product of glycolysis, the metabolic fate of pyruvate. So once you go all the way through glycolysis, you end up with pyruvate. Pyruvate is metabolized in one of 3 different ways, depending on how much oxygen is available in an organism, right? So you can have aerobic conditions. Under aerobic conditions, you're going to go on and you're going to make ATP. You're going to go through the citrate cycle. You're going to go through the electron transport chain, and you're eventually going to make CO2 and H2O, right? Then there are anaerobic conditions. Anaerobic conditions will make one of 2 different things. You can make lactate. This happens in our muscle cells. And it happens in those muscle cells during really, really strenuous activity, right? Really strenuous activity, where we're going to convert the pyruvate into lactate, right? And so that process is actually going to convert NADH into NAD+. And this is that enzyme we've been working on in lab, LDH, right? So that's one way that can happen, but there's also another way that it can go. You can go through ethanol production. So this is seen in many different microorganisms, right? Like Lactococcus lactis, which is a bacteria that produces yogurt, produces cheese, that kind of stuff. Or you could go the route of Saccharomyces cerevisiae, and you can convert pyruvate into CO2 and ethanol. And so that's how we make beer. And I'll tell you in a little bit more detail how that works. So what are our kind of fates of pyruvate in this sort of a diagram, right? So we're going through anaerobic here. We have glucose, and we're going to convert glucose to pyruvate. So we're assuming that happens. So either you can convert it into ethanol. So this is Saccharomyces cerevisiae, CREVESI. Something close to that. Or you can go through lactate dehydrogenase in our muscle cells, and you can make lactate, right? So, and then if you have oxygen present, you can go through the mitochondria, electron transport chain, citric acid cycle, right, all that, and end up with CO2 and H2O. And so a really critical function of pyruvate metabolism in all 3 pathways is that it is going to regenerate the oxidized form of the coenzyme NAD+. If you don't regenerate NAD+, see how we're regenerating NAD+, regenerating NAD+, and we are regenerating, we go through citric cycle, then we go through electron transport chain, and we make NAD+. So the whole point is that we're regenerating that oxidized form of the coenzyme NAD+. Why is that important? NAD+ is required for us to maintain flux through our glyceraldehyde 3-phosphate dehydrogenase reaction, right? We go from G3P to 1,3 bisphosphoglycerate. How am I going to make that fit in? Curve it. So you need NADH for those earlier steps, right? And if you don't have those regenerated NAD+ molecules, you're not going to be able to maintain glycolysis. So they're required. NAD+ regeneration is required to maintain our flux, right? Through our glyceraldehyde 3-phosphate dehydrogenase, right? That's here. So this regeneration can occur in the cytoplasm, right? In the case of lactate and ethanol production, right? But if pyruvate gets transported to the mitochondrial matrix, right, it gets fully oxidized into carbon dioxide and water by the citric acid cycle and oxidative phosphorylation, right? So then the NAD+ requires a mitochondrial shuttle system. And that shuttle system is linked to the citric cycle, the citrate cycle. And so we'll talk about that in a lot more detail when we get to that point, right? But the metabolic requirement for NAD+ in glycolysis can be seen in people who have defects in the enzyme lactate dehydrogenase, right? So this is a loss of function. It's not functional. And so you don't have enough NAD plus levels, right? And so these patients can't maintain in sustained intense periods of exercise because they have an inability to use glucose to produce ATP that they need for muscle contraction under anaerobic conditions, because if you're doing extreme exercise, you're under anaerobic conditions. And this can lead to fatigue, and in the most severe cases, it can lead to muscle damage, right? The cell's running out of ATP, and that's really, really not good. But you don't normally know it because you're not doing that much high intensity exercise. The general public isn't. And so you don't necessarily know that you have that kind of a mutation. The other thing that's really, really important, right, in terms of the fate of pyruvate is alcoholic fermentation, right? This has been done for hundreds of years in the production of beer, right? Because we know under anaerobic conditions, yeast are going to rely on glycolysis to make ATP. So how do they do this? What do they do? Well, in the process, right, the yeast are going to produce pyruvate, and then the pyruvate is going to be metabolized by alcoholic fermentation to regenerate NAD+. So if you kind of look, I don't know how much you know about brewing beer, but as biochemists, we love the science behind brewing beer, right? So we start with some kind of a carb source. It's usually a cereal grain, right, like barley, something like that, right? And we allow it to germinate. So that germination induces expression of things like alpha amylase, like maltase, right? All the enzymes that are going to be needed to break down the sugar. Then you have the mash, right? And so in mashing, you're going to break open the cells and you're going to release the enzymes. Okay. Now we add water to it and it turns into wort. Why they call it that, I don't know. Never looked that up. But so you have this water mixture, right, with your grain, and then you increase the temperature. The increase in temperature is going to optimize the catalytic enzyme activity, right? So all the enzymes are going to start to degrade the carbs, the proteins that are used as metabolites for the glycolytic pathway. All of those things are going to be created due to those activated enzymes. Then they're going to get rid of basically undigested cell debris, right? And then they take that wort and they boil it with some other ingredients. Sometimes those ingredients are like hops, something like that, right? And then you're going to cool it because it's hot at this point. And if you add yeast to the hot, you're going to kill the yeast. So you got to let it cool. You add live yeast culture, right? Now this is first added under aerobic conditions because you want to get the culture started. You want to get the yeast growing, right? But you only do it for a short amount of time. And then you immediately switch them over to anaerobic conditions so that they undergo fermentation. So those yeast are going to ferment the remaining sugars into ethanol and carbon dioxide. Now, big manufacturers are going to take that beer, they're going to filter it, and then they're going to inject carbon dioxide into it. They're going to carbonate it. But a lot of times those small microbreweries, what they do is at the very end, they filtered it. They do a extra injection of sugar and live yeast. And so it initiates a second round of fermentation right at bottling. And that actually is what carbonates the beer. So there are different processes depending on if it's a big company or a small company. But all right, so that is the end of chapter 9. All right, y'all have a great day.